know Dr. Misra? He was away for 2 years, but he's back now in the institute. Professor, clinical immunology, professor head. And if you need to learn something from him, one is his sense of humor, is something which has to be learned from him. And as a physician, he's extremely good. Even now, I think he hasn't forgotten his general medicine, which he practiced years ago.
Well, I don't know whether both of these factors contributed for Prithvi's selection of me.
Sir, you doubt your intelligence. That's the whole problem.
Things that apparently look so easy to you now in any discipline, a medicine or a surgery or radiology, which you just think or take it for granted, they're actually not so. The advancement of patient care by healthcare providers have in the last century, and of course we are very early into this century, so we don't know the audit. What have you been doing for the last, say, 10 years? Has it ever made any significant advances or not? It has definitely, the cake has not come on a platter. That's for sure. And what your basic, or I know you've been all kind of gone to medical school, then done some training in either a medicine or a surgery, and then setting yourself a very diverse discipline or super specialty. The idea of telling stories or events that have unfolded and have laid the modern path of our understanding is very important. Because as you see that some of these discovery were by logical deductions, maybe by hard work, some were chance discovery, and the basic point about you being a physician or you being a healthcare provider, you need to be very keenly observant yourself. Besides your skill, you have to have two factors so that you become something to deliver differently. I mean, lives of great men or lives of great discoverers have taught us one thing, that whom to follow. How would you change your life? You certainly wouldn't if you were to do the same thing. For example, as a DM student or an MS or MCH or someone in microbiology, radiology, you'll be doing a set work every day. The only thing that would distinguish you as something different is your observing skill. I mean, you look but you don't observe. Means you look and you don't see. And sometimes if you observe carefully, then you would think that something is wrong or something is different, and you can take examples of the stories that are there integrated with our development. I will be covering some aspect of transplant immunology. This is neither the history of transplantation and all, or it's an in toto description of the immunology history, which cannot be covered in a short. What I would like to tell you, how our concepts have changed based on people who made that change in concepts. Okay, and how events in the last century, the bigger event was what in the last century that separated it from all other centuries. What is a big event in the last century? If you take the entire 100 years and ask anyone, you probably were born at the late part of the last century. Something going. What was to a mankind? What was the thing that 20th century stands for?
Discoveries in genetics.
No. Nothing to do with science, nothing to do with medicine, nothing to do with what is 20th century known for?
IT tools.
Huh?
Information.
Huh? The two wars. The two world wars between 1913 to 1918. And the greater World War, the two. I don't know if you have ever seen any World War movies on the Greater World War II, and how much impact that has changed races, that has made discovery. Science would not be where it is had it not been for the wars. Okay, medicine would not have been where medicine is unless it was because of the wars. Some ethical experiment, some unethical experiment, something what the president wanted, that we need this to save our soldiers. All this would not have been possible. Lazy centuries don't produce activity. So we will say to it. Now the contributors to the history or failures of transplant has been by the transplant surgeons, by the tumor specialist, by the Mendelian geneticist, and the basic biologist. There was nothing called an immunologist at that time. Now, since the history is to do with transplant, the surgeon has to come in, or the group of people who ever tried or attempted to transplant a part of the body, cells or tissues, into a genetically distinct individual or a different individual. Now the earliest history actually credited to one our guru called Susruta, you know that. In surgeons, if you know that, probably in 324 BC, he did a surgery by taking skin from some part of the same body and reconstructed nose. Any guess why a nose would be cut? Why your nose is not being cut, and why at that time nose were being cut? Punishment. No. War, human society has been known to fight and all that. Maybe for punishment, but this use of sword has led to inflicting other injuries, and one part of the body that was frequently non-lethal punishment was to cut the nose or the ears chopped off. We don't know, but he did it from patients or that victim's own skin and succeeded. There are many instances from other countries also around the turn of at the birth of Christ that it is possible to reconstruct nose or ears even. Nose was easier. In the end of 19th century, surgeons in France and Germany, they were busy. These are called experimental surgeons. I mean, beside the skill that they have for doing on human beings, these experimental surgeons transplanted kidneys between dogs. There are reports also from a place called Lyon in France. Dr. Jaboulé tested pig and goat kidneys, transplanting one with some limited success. Suppose a particular animal has renal failure, transplanted kidneys from other one, because the other person the other animal had two kidneys. So one kidney could be placed at various places, from the neck to the leg, or at its own position in the pelvis. And they did find limited success. From France, Alexis Carrel was awarded the Nobel Prize in Medicine in 1912 for acquiring a skill of anastomizing vascular structures, and he grafted a dog's head into the neck of another dog, for which he was given Nobel Prize. I mean, not many surgeons are given Nobel Prize. You do wonderful surgery, but no. In 1912, he was given Nobel Prize for grafting or developing a skill of vascular anastomosis. How can you from a transplanted structure or the graft, how can you suture the vessel into the host vessel? However, these from Ukrainian surgeon Voronoi, he did the first human cadaveric transplant in 1933. Dr. Gutheri, who was working with Alexis Carrel, he commented that though the skill part is there, invariably the graft failed. Means all these transplants from various experimental or even from a cadaver into a diseased human being, it didn't succeed. And the ultimate result was rejection. So he commented that the outlook is by no means hopeless. Of course, by that time there were about more than hundreds of cases have been done, and all have ended in failure. And he thought that now it is beyond the surgeons. In fact, the so-called zeal for transplant that gradually was dying down. I mean, no one did transplant, except for one transplant that was somewhat alive, and that is the corneal transplant. Corneal transplant is a different cup of tea, because cornea is a vascular structure. Any vascular structure transplant failed, and he thought that the principles of immunity, what is basically the body doing, the host or the recipient is doing, is rejecting the tissues. And immunity is something to do with the defense. And that we have to explore that if we were to gain success in transplant. And by that time, several diseases were controlled by immunological principles. They were not called immunologists, but you would know that Pasteur is known for vaccination of cholera, anthrax, rabies. We don't see rabies or that to that extent because of these people who produced vaccination. I mean, besides Edward Jenner. Edward Jenner is earlier, 1 century old. Early antibodies to antigen. Cox from German, he described tuberculin hypersensitivity. People were rushing to his clinic to get an extract of tubercle bacilli, what we now know as the Montu test, that they could be protected. And similarly, Baudet and Guingo, they had described the complement activity. Various immunological phenomena had been described, which was there in the human body. And not only that, the practice of this has helped people. We are talking of pre-antibiotic era. 1945 was penicillin first described at St. Mary Hospital in London. And you can see that why Dr. Guthrie thought that maybe we need to be aware or somehow capture the development of immunology to help prevent rejection. All these immunological developments were based on the body's capability to produce an antibody. These are all based on humoral principles. Now the contribution of the tumor surgeons were they thought that if microbes can be prevented by giving a part of the microbe to the host, why not naturally occurring tumors, you take few cells and give it to the host, and the body would develop an immunity so that the larger mass can be tackled. So, taking an idea that with infectious disease, success of infectious disease control in the society, the tumor surgeons, they were busy transplanting tumors into animals. Whichever tumors, and tumors are usually immortal cells. Tumor cells, usually you can grow them and they will not die. They have immortality. And several tumor cell lines were injected into animals to see that the animal would develop an anti-tumor immunity. But none of these experiments were successful. They were nothing like an anti-tumor antibody in the same as an antimicrobial disease. So, but this led, for those who don't know the terminology, between identically similar individual, a transplant is called an isograft. That is a monozygotic twin lending of tissue to its twin. If it is from the same individual, it's called the autograft. From a genetically distinct individual, it's called an allograft. And between when the tissue or the organ is taken from across the species, it's called a xenograft. It's terminology. There were about these 500 cases, 500 reports of tumor immunologists trying to raise the tumor immunity, and George Kohn, a German, in 1912, he wrote a book. He said, enough is enough. Let me gain all the experience of different people. And he propounded what is the law of transplantation. And it says that xenogenic transplantation invariably fails. Rule number 1. Rule number 2, allogenic transplants usually fail. Allogenic means in the same species but genetically different individuals. Autografts are almost always successful. The initial take of a first allograft, the first allograft is taken first and then it fails. And the second graft fails faster. And graft success is more likely if the donor and recipient are blood relation. This was his book. This was the law of transplantation as translated in English. It was all in German. His book was in German. 4 years later, the rule, because of some more publication, the rule slightly changed, or they were revised. And something more was added. The sensitization to the rejection requires some living cells. And in the rejected graft, the lymphocytes predominate. And there is no tissue specificity, means all living tissues invariably fail, which have vascularity in them. The whole blood, if it is vast RBC, they can be immunized without problem, and passive immunity cannot be done by serum. Like the success of the previous century of infection, you can't do something like whomever you want to, the recipient, if you give some part of the donor cells, allow the cells to have an tissue immunity, that is not possible. And the contribution by the geneticists who were working with usually inbred animals, they thought in these animals you do brother and sister mating so that you have truly inbred animals. Something came out, and those were in 1903, Jensen first albinomized that genetic differences control rejection. And in late 1930, Peter A. Gorer, he was a British gentleman, he was injecting rabbit RBC into, as mouse RBCs into rabbit cells, rabbits, and testing for anti-mouse RBCs immunity. And at the same time, there was another person whose name was George Snell across America. He was in he also was doing similar experiment, and they observed that multiparous pregnant females, they have in their antibodies which were directed against the lymphocytes, not against the RBCs, and sometimes these can cause problem with transfusion. They combined, and this is genetically controlled, and they described for the first time the mouse system of HLA, which is called as H2, because he had described H and he had described 9, and they combined and they found 9 alleles. And of course, then came the World War II. After 1930, transplantation, those initial activities, they stopped. Then came the war. so happened a young Oxford zoologist who was doing some research in a hospital called Magdalene Infirmary in Oxford. A British plane crashed, and the pilot was suffered from about 80% burns. And he was visiting the hospital. This gentleman, Peter Medavar, is a young zoologist. He had got nothing to do with patient care. He was going to the burn unit to get some skin tissues and all that. Then he observed that he was very emotionally, because being a British national, a British plane was just 200 foot from where he was living, 200 feet or 200 yards. And the story goes that he went to the hospital and saw that the pilot was suffering 80% burn. So he suggested to the surgeon that let's put someone else's skin on it. The rest of the skin, the area from where his own skin were grafted, they succeeded, but someone else's skin it didn't. And he was so moved, and he observed that the second allograft, so he said that there is something wrong, and he applied for a research grant to a committee during the war, which is called the War Wound Committee. The British Medical Council, like our Medical Council of India, British Medical Council were busy taking the help of doctors to cope up with the wounded soldiers. So to the War Wound Committee, he applied for a grant to work on this phenomena of allograft rejection. He moved to Glasgow from Oxford, and he did some carefully planned experiments in mice. He's a zoologist, so it was easy for him to do things in animals. Then he made a very valid observation that the accelerated regression of the second set of homograft means that there is some systematically the body is fighting. It's not just the local, that the vasculature or something. Most people would think that something happened locally. That skin, the underlying tissues were non-vital. They were not well nourished. But he said, no, it's the body is the entire body of the host or the recipient is trying to prevent this. And by looking at those rejected samples of tissues, he found that they were having lymphocytes. You would be surprised to know at that time, lymphocytes was not given any importance. Two cells were important in our peripheral blood. It was the RBC and the leukocyte. The leukocyte was thought of as fighting against defense, and the RBC's function was known. And he said instead of leukocytes, it was the lymphocytes that was there. But then the general knowledge about lymphocyte was so poor, because they thought that lymphocyte, the cytoplasm is devoid of cytoplasm. So it can't be a very functionally active cell, because the cytoplasm of the lymphocyte is just a rim. He and his postdoctoral, he moved back to London, Medavar. He moved back to London, and with two other giant figures called McFarlane Burnett and Fenner, they observed that this transplant rejection is to do something similar to delayed type hypersensitivity. Delayed type hypersensitivity is what we get as a Montu test. The reaction that we get as a Montu test is called a delayed type. And Mitchison in US could produce that a similar kind of rejection by passively transferring lymphocytes. So in allogenic tumor situation, so that you know that they now thought that it is a lymphocyte which is doing the funny, is responsible for transplant. At that time, lymphocytes' importance were not known. Though it was something around 1940s to 50s, just after the World War 50s, what Billingham, Sprint, Medawar, and of course with the idea of McFarlane Burnett, what they had observed that if some lymphocytes were given to embryo, subsequently you do a graft, that is not rejected. The whole war or the whole fight was how to protect a transplanted tissues, the means of protecting it. Though the lymphocyte role was not clear, they were not able to understand what the damn thing is doing, but by these kind of experiments, McFarlane Burnett was later knighted, gave his theory of self and non-self. It was happening, everybody was observing that. But he gave a theory that between self and non-self, and that this self and non-self is decided much earlier in the life. So body has a capability not to react to structures, protein, antigens, if it has been exposed in the embryonic life. You got my point? After the chicks are hatched, the embryos are hatched, if you give them something, they will reject. If you can manipulate an embryo and give something distinctly genetic material, it will not react after birth. So the self, that was McFarlane Burnett, the self-non-self theory. But Medawar continued and he said that, can I prevent allograft rejection by manipulating animals? So actually he Brent and Billingham, they did work and they did transfer lymphocytes into chick embryo and managed to show that it is possible. Subsequently, if you do a transplant, it is accepted. Because of this sorry. Clearly, the lymphocyte functionality was increasing. No longer it was thought to. Many people thought that lymphocyte at that time was a precursor cell of other hematopoietic cells. These Govans, he did the radio labeling of the lymphocyte, and he showed that the lymphocytes from the lymph node duct actually goes out of the lymphatic channels, and therefore it must be doing something. Paul Terasecki, he was a postdoctoral fellow under Brenner, under Medawar, and he observed that if you give small lymphocytes from someone allogenically distinct animal, that lymphocyte can cause some disease. It was the other way around. The graft can cause disease in donor. And Simonsen from Copenhagen is the discoverer of the first graft versus host disease. When he injected adult lymphoid cells into chick embryo, the embryos, they died of severe hemolytic anemia, splenomegaly and death. So it's a bilateral thing. Not only the recipient rejects the graft, The graft also, if it has cells, the lymphocyte, it can cause problem with the donor. And it was clearly obvious in the early part of the 50s that the lymphocyte is playing havoc with rejection. And then came this test, which test actually stood the time. So there was no test that which transplant would succeed. So this test, what this last group of authors, the Bain, Das and Weinstein, they described that if you culture lymphocyte between two genetically distinct individuals, the lymphocyte becomes larger, divides and proliferates. Both lymphocyte means you take from A, mix with B, artificially grow them, and these lymphocytes are undergoing change. He called them lymphoblast, means the cells which are small, they acquire more, the nucleus becomes bigger, the cytoplasm showing protein synthesis. And if you stop one of the cells, the DNA from making protein by an anti-cancer agent called mitomycin, you can test whether the donor and the recipient, their cells can match. So these mixed lymphocyte culture were to be taken as an in vivo test before actual transplant. Let us mix lymphocytes. If the lymphocytes won't have blast transformation, then the graft would succeed. When there is a question of donor graft between distant blood relative, you are finding very difficult to keep your eyes open. It's a bad part of maybe. Anyway, this kind of simple test was to be adopted, and as we now understand, this was a test for HLA mismatching. No one knew at that time. There was this H2 gene was known, but how to know that it's like we test for antibodies against blood group A or blood group B before we transfuse blood, and before we transplant, before then they realize that before they transplant tissues. And of course, it was not only, now we know that it is not only important for tissues, it also is important in immune system, this molecule. Subsequently, Zinkernagel and Doherty got Nobel Prize when they described that it is your own HLA that can fight against, means we all have different HLAs. So our body is only capable to fight infection if our own cells, HLA cells, are fighting it. We cannot with someone else's HLA. Body cannot allow that. Anyway, so and then came subsequent development in the 60s when Miller from London and Good from Minneapolis, they did neonatal thymectomy in mice, and the result was that there was severe immunodeficiency, lymphopenia, wasting syndrome. So the importance of a small gland in the neck was there, thymus. In adult, as we know, that thymus is atrophied. But it is that small gland that if you take it out when you are in an embryonic life, particularly in animals, these animals die. And it was just a chance discovery by Bruce Click from the University of Wisconsin. He was a veterinary scientist. If he removed a cloacal organ called bursa fabricus, the animals would again, the chicken would have no antibody. So those discoveries led to find that these lymphocytes are not one, but there are two types. The T lymphocytes, which have been originated from the thymus, and the B lymphocyte, which have been kind of the bursa fabricus is important, and therefore the name B. B means bursa fabricus. And now we know that B cells are responsible for antibodies and T cells are responsible for cell-mediated immunity, and there is an intense cooperation between the two. But this was not known when the transplant people were actually busy doing mixed lymphocyte cultures. It is the and the lymphocyte also came important. Then people started looking at some earlier studies by Rouse and Murphy. They had described in 1912 that these, when I told you in the beginning, they were busy transplanting tumor cells into animals, that if these animals were given to embryo, till that time the tumor cell line survived. But as soon as the embryo hatched and lymphocytes appeared in circulation, the tumors were rejected. In 1945, Ray Owen, he observed he described a very important thing, that in cattle which have got dizygotic twins, each of these twin pair have their own placenta. Somehow the placenta is joined together and allowing a common blood circulation between the two. Even if there was a female embryo and a male embryo, there would be circulation. And he observed that there is a chimeric state possible. Dizygotic means each has its own placenta. Monozygotic, each have the same placenta. So dizygotic twins are genetically distinct. And one can be a female, one can be a male. But he observed that even if in the adulthood, he could see that some RBCs of the partner is circulating, the body is not getting showing. So Burnett propounded the theory that I told you, that if there is an exposure during the embryonic phase, body will tolerate the tissues. For their work, both Medawar's work, the zoologist, and Macfarlane Burnett was awarded the Nobel Prize in Medicine in 1960. Medawar felt very sorry because his other two students or colleagues, Billingham and Grant, also worked, and they were given the prize. So he felt very sad about it. But he persisted for 30 years to prove that cells are involved in rejection, and it is possible if you inject some cells of the donor at the embryonic stage to the animal, tolerance is possible. So this is one of a great event in transplant immunology. Around 1954, that was the year where Joseph E. Murray did the first renal transplant. And for this piece of work, 38 years later, he was awarded the Nobel Prize. But he did this transplant in an identical twin, not requiring any immunosuppressor. He succeeded because it was, he did it in Boston, in the Brigham's Women's Hospital. As you know, it is perhaps the best medical school. Boston Medical School or Harvard Medical School is the best, and the hospital attached to it is the Brigham's Child and Women's Hospital. He did it there, he was surgeon. There were 30 surgeons in that department, and all said that it would fail, because all transplants had failed by that. But with these arguments of McFarlane, Burnett and Medover, Morey, he was the hero. He thought, no, because they are identical twins, and the transplant succeeded. So much it succeeded that one of the transplanted, the recipient, she later on actually became a mother. And so the Nobel Committee felt that that is a significant advancement in medical science. And he also said that he went on to do 12 more transplants. With taking risk, next time transplant would be slightly distant relative than exactly identical twins. But he observed that there is rejection if they are not identical twins. Organ, kidneys transplanted across dizygotic twins failed. But he felt that by immunosuppressive drug, it is possible to win this and then between 50s to the 2000s, what we saw was people trying how to knock immune system with transplant. In 1950s, they were doing whole body X-ray irradiation, but it was sometimes have significant side effect and morbidity, and some of the people actually died. In 1959, Sord and Ramesek, they found that 6-mercaptopurine, it inhibited antibody. Roy Can, he discovered the imidazole derivative of 6-mercaptopurine, which is as such a very toxic drug. 6-mercaptopurine is used in tumor situations, if you know. It causes a plethora of side effects, including mucositis, vomiting, immunosuppression, bone marrow suppression. And they said that they called the drug azathioprine, and that was less toxic. And Billingham noted that daily administration of cortisone was discovered around 52 or so, 46 to 52, for which Philip Hench got the Nobel Prize in 1956 for trying cortisone in rheumatoid arthritis patients. And Stargell, he said that if there is rejection and you give large dose of cortisone, it can reverse rejection. And because there was this notion that lymphocyte was the thing that was rejecting, Waxman and Woodruff tried anti-lymphocyte globulin to prevent rejection, and Stargell later on said that anti-lymphocyte globulin can save kidney graft, which we know is correct. So the second half, between 50s to 2000, the second half of the last century, there were number of immunosuppressive drugs were developed, and you know that in the 1980s cyclosporin came, then tacrolimus, then came mycophenolate mofetil, rapamycin, and we have antibodies to various lymphocyte surface membrane receptors, because the lymphocyte is doing the trick or is responsible. And much of our success of transplant is because of judicious use of these drugs and matching the donors as much as we can possible. So much, and you see that there are people from scientists from various disciplines, surgeons, immunologists, experimental geneticists, all have contributed to the success story of transplant. So they all thought that the second part of the half century, a new science have come into existence, which is non-sectorial. Sectorial means you practice gastrosurgery, you don't know what is happening in endosurgery, you are remaining there. But for the success of transplant, you need expertise from various disciplines. And so much so that a new society was formed called the Transplant Society, and the New York Academy of Science, which was one of the leading science professional group, arranged 6 transplant societies meeting between then and the last one probably was held somewhere in '84. And people who would come to that society, geneticists, zoologists, that society which saw knowledge in each area kind of converged to make transplant possible. Of course, similar things were happening in the HLA system, and for their description of the genes and the proteins, George Isnell and Jean Dowsett were given the Nobel Prize in 1980 for the H2 H2 gene, that is the gene in the mouse, and the human gene is called HLA, the similar gene. So now in these societal meetings there were one debate. The debate was whether we shall do more basic research, look at more antigens that were not matched across genetically to distinct individuals, or to work for more better immunosuppressive drugs. So whether to invest money in basic research or to invest money in newer drugs. And as we have known now that in the next 50 years, both argument is correct, and the success story of transplant and of immunology is possible because our knowledge of the genes responsible and the proteins responsible and the effector cells, their cytokines, that knowledge, the basic work in that has helped to develop targeted anti-transplant or anti-rejection therapy, and of course newer drugs that would be more targeted and like anti-cytokine targets. So both views were correct, and so in the capsule that is the transplant story is a success story of the last century actually. Little late.
Usually less time to speak as a group.
You have 1 hour to speak.
And try to finish it early.
So thank you.